It's my honor to introduce our second speaker, Komal Yasmin. So Komal started her academic journey back in Lahore in Pakistan, and actually she started her epigenetics journey during her master's studies in Pakistan, and then she acquired this quite competitive scholarship from Wellcome Trust and moved to University of Oxford to Neil Brockdorff's lab, where she has been studying the CpG Island methylation during inactivation in mouse. Also, she's currently searching for postdoc positions. So if you are lucky to get an application from her, just look out. Floor is yours, Komal. You can start.
Thank you so much for that kind introduction. Let me turn my screen share on. And if you could just confirm that you can see my screen. Okay, so I'm assuming my screen is turning out fine. Thank you so much. And thank you so much for inviting me to the Fragile Nucleosome seminar series. I'm very happy and honored to be here. Thank you everybody who joined. And today I'll be presenting a part of the work that I have done during my DPhil in Neil Brockdorff lab at University of Oxford. So at the Brockdorff lab, we are very much interested in studying a developmental process called X chromosome inactivation. So X chromosome inactivation is a dosage compensation mechanism in which one of the X chromosome in female mammals is transcriptionally silenced in order to equate the dosage of X-linked genes between male and female mammals. And this process is initiated by the expression of a long non-coding RNA X-cyst, which spreads along the length of the chromosome and brings about transcriptional silencing, ultimately leading to the heterochromatinization of the inactive X chromosome. And one of the late stages of X chromosome inactivation is called CpG Island methylation. So CpG islands are the CpG-rich regions, which are found in association with about 70% of the mammalian genes, and their methylation is correlated with the silencing of the associated genes. In mammals, this process is catalyzed by enzymes called DNA methyltransferases, abbreviated as DNMTs. And in mammals, there are two DNMTs that are involved in establishing CpG methylation called DNMT3A and DNMT3B. Now, both DNMT3A and 3B are highly homologous proteins that are very well conserved, as you can appreciate from the schematic here. Not only do they have the same domain composition, but they also share the linear order of these domains, and understandably, they work redundantly across most of the genome. An exception to this rule, however, is the inactive X chromosome. And a previous study from our lab has shown that the removal of DNMT3A has no impact on CpG island methylation on the inactive X chromosome, while the removal of DNMT3B results in the complete abrogation of CpG island methylation on the inactive X. And during my DPhil, I was very much interested in finding out why does DNMT3B exhibit this specificity, and why is it specifically required for establishing CpG island methylation on the inactive X chromosome. So in order to answer this question, I'm making use of an experimental cell system, which is mouse CS cells. And in our lab, these are very special because they have been derived from an F1 generation of a cross between two very different mouse strains. And what that means is that when we analyze genomics data from these cell lines, we can actually discriminate between the two parental alleles based on the single nucleotide polymorphisms, or otherwise known as SNPs. These lines have been further engineered such that on one of the X chromosome, the X-cyst promoter has been replaced by a doxycycline-inducible X-cyst. And what that means is by the addition of doxycycline, we can actually induce X-cyst expression and i.e. have an inducible X chromosome inactivation. Now, the CpG island methylation on the inactive X chromosome has been shown to be specifically dependent upon differentiation. And for that, what I do is that I remove a media component called LIF. And LIF is generally required for maintaining ES cells in their stem cell state. And so when I do that, the cells spontaneously begin to differentiate, and this process is called embroidered body differentiation. And I can then sample cells while this differentiation is happening and study the CpG Island methylation status. So in order to study the CpG Island methylation, I have made vast use of an assay which is called MBD affinity purification. It's also known as short form MBD-seq. And this technique gets its name from the use of MBD proteins, which is an abbreviation for methyl binding domain. And these proteins have an affinity to bind to CpG-rich methylated DNA. Now, during this protocol, I conjugate these proteins with magnetic beads. And what that allows me to do is to carry out a very straightforward IP reaction in which I can extract and fragment the genomic DNA from my cells, perform an IP with MBD protein where I can pull down the CpG-rich methylated DNA, and I can then elute that DNA and use it for next generation sequencing. So to give you a brief idea, the data from this experiment looks like this. In the red track, you can see the peaks, which represent CpG-rich methylated DNA. And as I've mentioned before, because of the way the cell lines have been derived from two different mouse strains, we can actually computationally split this data into the two parental alleles. And in my case, I'm very much interested in comparing the active X chromosome with the inactive X chromosome. And so this technique gives me a very, this technique is a very powerful method of studying CpG Island methylation on the inactive X chromosome. So now that we have the tools introduced, coming back to the question of why does DNMT3B have the specificity for the CpG Island methylation on the inactive X chromosome? The very interesting thing is that although we have known this for a while, we actually do not know how DNMT3B contributes to this process. And one obvious hypothesis, of course, is that DNMT3B catalyzes the CpG Island methylation in itself, since it's an enzyme. A second probability, however, is that it serves as an accessory protein and helps to activate other methylating complexes. So what do I mean by serving as an accessory protein? So previously, I have shown you this schematic of DNMT3A and B, which is an oversimplification. In reality, both DNMT3A and 3B have multiple isoforms. However, a feature unique to DNMT3B is that it has a number of inactive isoforms, in addition to the catalytically active isoforms. And these inactive isoforms have very important regulatory roles in CpG methylation. So one of these, for example, DNMT3B3, is very well characterized for its ability to heterodimerize with the catalytically active DNMT3B and 3A, and helping them to enhance their catalytic activity, as you can see here. A recent structural biology study has shown that DNMT3B3 has an ability to bind to the nucleosome acidic patch, which then allows it to position the catalytically active 3A or 3B, in this case, I'm showing you 3A, to be positioned on the linker DNA where they then carry out CpG methylation. So with this background in mind, what I've shown you so far is that DNMT3B is very important for CpG Island methylation on the inactive X chromosome, but we actually do not understand how does it carry out CpG Island methylation. So one idea is that DNMT3B catalyzes the reaction itself, in which case, if I remove DNMT3B, I would be able to remove all the CpG Island methylation from the inactive X chromosome. And I would be able to fully recover all of this if I only introduce the catalytically active isoform of DNMT3B. The second model, however, assumes that DNMT3B serves as an accessory protein, in which case the major or the main candidate would be this catalytically inactive isoform DNMT3B3, which would then heterodimerize with DNMT3A and 3B, allowing them to anchor to the chromatin where they could then carry out CpG Island methylation. So these are the two models that I wanted to test next. So, but before I go into showing you the data, if you look at the MBD-seq signal during EB differentiation, this is how they look like. So you can see a very nice gradual rise in the level of CpG Island methylation as the differentiation progresses. So here, every plot is showing the average MBD-seq signal on all the CpG islands on the X chromosome overlapped on top of each other. And I'm showing you the relative gain of CpG island methylation on the inactive X chromosome in comparison to the active X chromosome. If we then look at the DNMT3B knockout line, like you can see here for the two clones, what you see is a complete loss of CpG island methylation. So these lines have a very nice, they form a very nice baseline on top of which I could then introduce one isoform or the other, and then see if I'm able to recover the CpG island methylation. So if we focus on the day 12 time point, what I've shown you is that in the wild type cells, we can see a rise in the CpG island methylation on the inactive X chromosome, which is completely gone in the case of DNMT3B knockout line. If I then only introduce the catalytically active isoform of DNMT3B, I see a complete recovery of all the CpG island methylation on the inactive X chromosome. If I, however, only introduce the catalytically inactive isoform of DNMT3B, I do not see any rescue of CpG island methylation. So what that shows us that out of the two models that I proposed earlier, we can completely exclude the contribution of the catalytically inactive DNMT3B3 in the process, bringing us to the catalytically active DNMT3B carrying out the CpG island methylation. But this complicates the problem a little bit, because you remember me telling you that DNMT3, the catalytically active isoforms of DNMT3A and 3B are actually very similar enzymes. And the question then is, what imparts DNMT3B this specificity? So despite the close similarity between the two proteins, it turns out that the N-terminal domain of DNMT3A and 3B is almost unique to the homolog, with almost no similarity between the two proteins. And so I next hypothesized that perhaps the N-terminal domain was the specificity imparting region of DNMT3B. And it could be functioning by mediating its interaction with DNMT3B specific interactors, which could then be specifically recruiting it to the X chromosome. And if that is the case, then theoretically, if I truncate the N-terminal domain, I would get rid of these DNMT3B specific interactors, and as a result, also get rid of the CpG island methylation, despite the fact that the catalytic domain or the catalytic function of DNMT3B is completely intact. So this is exactly what I tested next. And indeed, when I complemented my DNMT3B knockout line with the N-terminal truncated DNMT3B, what I found out was that the CpG island methylation rescue that I observed upon the full-length DNMT3B was indeed gone upon the removal of the N-terminal domain. And what was quite interesting is that this effect was not only limited to the X chromosome, but all the CpG islands and the CpG-rich regions that were captured in my MBD-seq data that were dependent upon DNMT3B for their methylation failed to get rescue in the N-terminal truncated DNMT3B knock-in line, while the DNMT3B independent CpGs remained unaffected. Now, this brings us to the question, what does the N-terminal domain actually do? And interestingly, the N-terminal domain of DNMT3B is the least of all the studied domains of the N-terminal domain, of the N-terminal protein. So we do not yet have a full structure of DNMT3B. And on the basis of the amino acid sequence alone, it is predicted to be completely unstructured. And that is quite interesting given the fact that the N-terminal domain of DNMT3B is very well conserved among different mammalian species. And this is quite interesting given the fact that it has been shown to be completely dispensable for its catalysis. So my original model assumes that the N-terminal domain is somehow mediating the recruitment of DNMT3B to its target loci. And this is what I tested next using a ChIP-seq. And what I found out was something very interesting. And overall, what it seems like without the N-terminal domain, I'm depicting with the delta N signal here, the DNMT3B behaves very much similar to its full-length version shown here as wild type. And overall, they have a very similar genomic localization in the ES cells. However, if I perform a ChIP-seq for DNMT3B in the differentiated cells, what I can see is a very profound loss of localization on the X chromosome in differentiated cells. So what this shows us is a very context-specific role of the N-terminal domain, which becomes apparent in differentiated cells, especially on the X chromosome. I then also took a closer look at the CpG islands, where I was able to see a loss of the residence of the N-terminal truncated mutant of DNMT3B on the CpG islands. And this was true for the CpG islands on both the X chromosomes as well as the autosomes, which correlates very well with the MBD-seq data, where I showed that both the CpG islands on the X chromosomes and the autosomes were equally affected by the loss of the N-terminal domain. Now, what is still not clear to us and what should be determined next is the mechanism where which the N-terminal domain actually functions to mediate this localization of DNMT3B. And an important consideration to keep in mind is the role of differentiation in this process, because it appears that something is happening during this developmental time window, which allows DNMT3B, which is expressed in ES cells, to then specifically go and get recruited to its target loci in the differentiated cells. And this is very interesting, given that this is the time window at which the levels of DNMT3B actually begin to taper inside the cells. So two of the obvious models that come to mind via which the N-terminal domain of DNMT3B is mediating its recruitment is, as I proposed earlier, is that the N-terminal domain is the docking site for the DNMT3B specific interactors. These interactors could be becoming available only during differentiation. And so it's important to understand what are these DNMT3B specific interactors that interact with it only upon differentiation. And then it'll be interesting to see if this interaction indeed is dependent specifically on the N-terminal domain. Another possible model is that the N-terminal domain is important for reading certain chromatin modifications that become enriched only during differentiation on the target loci for DNMT3B. And in the case of the X chromosome, for example, these are very well characterized, and these are polycomb-mediated ubiquitination of the histone 2A and H3K27 trimethylation. Another chromatin modification that gets enriched on the inactive X chromosome is the H3K9 trimethylation. So all of these can be candidates to be studied for a probable kind of reading of the N-terminus of the DNMT3B. So these are a couple of experiments that I'm interested in next, and hopefully they will give us some clarity on how the N-terminal domain is functioning to mediate the recruitment of DNMT3B. And so with that, I would like to summarize whatever I have shown you so far. So I have shown you that the DNMT3B is critical for CpG Island methylation on the inactive X chromosome. I have shown you that the catalytically active isoform of DNMT3B is required for the establishment of CpG Island methylation on the inactive X. And the catalytically inactive isoform of DNMT3B, i.e. 3B3, has no contribution in the process. I've also shown you that the N-terminal domain of DNMT3B is very important for its role in the establishment of CpG Island methylation. I've also shown you that DNMT3B has an important role in the localization of DNMT3. The N-terminal domain has a very important role in the localization of the DNMT3B. However, this role is very context-specific, and it becomes quite apparent in differentiated cells for its localization on the X chromosome. And it also appears to be very critical for the localization of DNMT3B on the CpG islands. So that's all that I've shown you today. And with that, I would like to thank my lab, the Wellcome Trust, for funding me, and all of you for your attention. And of course, the Fragile Nucleosome community for inviting me to the seminar series.
I think you are muted.
Oh, sorry, I have been talking for no one. So thank you, Komal, for this amazing presentation. And it was nice to see this amazing flow of data and story from you. We already have 2 questions in the Q&A box. One of them is from Jason Fan. Jason asks, hi, great talk. I was wondering how different the N terms of DNMT3B1 is from 3B3, and if you think methylation rescue would occur if you swap the N terminal of 3B1 into either 3A or 3B3 rescue under the 3. When you knock out 3B and you just swap the N terminal to a 3B3 or 3A, would you see this effect? This is actually my question too.
So the N-terminal domain of DNMT 3B1 and 3B3 are exactly the same. The isoform DNMT 3B3 is only lacking a short fragment somewhere downstream. So the N-terminal domain is exactly the same. Plus, what my data shows is that DNMT 3B3 has no contribution in the establishment of CpG Island methylation at all. So, I mean,
even swapping like with 3A, or wouldn't you think that it is it wouldn't work, or like
so that's a very interesting question, and similar kind of swap experiments have been done very thoroughly in DNMT between DNMT 3A and 3B, and it has shown that the proteins actually tend to lag. I have done a similar experiment where I've swapped the N-terminal domain of 3B on 3A, which I didn't show in the interest of time, but it turns out that that does not let DNMT 3A behave like 3B. So there is more to this story than just the N-terminal domain.
Okay, interesting. So the second question is from Carolyn Brown. Carolyn asks, with respect to the role of chromatin marks versus differentiation specific factors, the availability of them, have you examined cells with DOX induction of cyst without concomitant of differentiation? Concomitant differentiation, sorry.
So that's a very good question. And of course, if it were possible to get CpG Island methylation without differentiation, that would remove one confounding factor, and that will simplify the experiment. But unfortunately, it has been very well established that you definitely need differentiation in order to have CpG Island methylation. That is that's a whole another story that our lab has published, and it's because of a protein which is called SMCHD1, which only comes on the inactive X chromosome during differentiation, and CpG Island methylation seems to be very nicely correlated with this protein. So unfortunately, it is impossible to study CpG Island methylation on the inactive X chromosome without differentiation. I hope that answers the question.
And the last question is from Sandeep. Sandeep asks, very clear and thorough analysis of the NAMS3B role on inactive X. I agree. And I wonder if you have examined gene expression from the inactive X in the absence of the NAMS3B in your hybrid female embryonic stem cells of an induction of cyst RNA. Relatedly, is it possible that the NAMS3B is recruited to the inactive X by cyst RNA itself?
Very, very good questions, both of them. The first question about examining the gene expression, and I should have probably mentioned it, that with all of these experiments in parallel to the MBD-seq, I also did chromatin RNA-seq. And this was to make sure that the X chromosome inactivation was going very well and the differentiation was going very well. So without DNMT3B and without CpG Island methylation, the X chromosome inactivation goes on just fine. So DNA methylation or CpG Island methylation in itself is not required for bringing about transcriptional silencing. And the second question I assume was if DNMT3B is recruited by X-cyst itself. So this question too has been addressed by some very nice studies which have looked at the interacting partners of X-cyst. According to my knowledge, DNMT3B is not one of them. So
thank you. Thank you. So this is, I think, the end of the questions. And thanks everyone for joining today and for this really amazing seminars. Hope to see you in 2 weeks' time.